And welcome back. Okay, now we're in these last very short classes. Each of one has a few minutes or up to 20 minutes most. Just showing you applications that maybe you didn't know, right? And trying to fix some of the concepts we had so far. So now we're talking about applications in agriculture. And believe it or not, there's quite a lot, right? I've told you about food sterilization and everything, but there's more than that. One of the very important things is radioentomology, meaning you try to use radiation to understand how animals uh, work, how animals live, and the whole system, right? This is used for plague control. It's an economic alternative to pesticides, safer, and for instance, it's used for male sterilization. And just as a side note, in the last years, it has been used not only for uh, agriculture, but only also for health. There was a study trying to use these same techniques to male sterilization. Basically, you sterilize the male and release them so that they compete with healthy males, with males that are not sterile, and they start to, as they compete, they reduce the number of males, and then they reduce the amount of some kind of problematic animal. In this case, we're talking about the the insect that causes dengue fever. It causes another illness called chikungunya, and it has been a very serious problem in Brazil for many years. Not only Brazil, but lately Brazil has been under serious stress because of dengue fever. And there was some years ago something for Zika and chikungunya, which are also spread by the same insect, which is called Aedes aegypti, and its friends. There are some cousins to Aedes. I forgot the name of the other ones. So if you can sterilize male, these sterile males will take the places of the other males, and you will probably reduce a lot the population of these insects, right? One example, for instance, they used in Mexico and the United States to eradicate the blowfly. This is much cheaper and essentially much more effective than pesticides, because pesticides you only get the ones that are living. If it's an egg, you won't get it, and you have more issues there because sometimes you have migrations and everything. What you have here, right? You can irradiate the plague in the whole region if you use it correctly. What we won't do with pesticide. The idea, you have a bug, for instance, you mark the bug with a radioisotope, and then you will see which of the other bugs there is the predator for that bug. Because if this one is marked with radiation, the one that is the predator will show the presence of that radiation, and the one that is not a predator will not show that. And then you just get the one that you do this in the lab, and then you have some population of this predator to the region you want to this bug to be eliminated, right? This technique in general, it's also used in industry and many other places. It's called radiotracer, meaning you use radiation as a tracer, right? You put radiation at some point in your system. This point may be a place in your oil tubes, maybe a place in the water system, and maybe a bug. And you just follow the radiation, and by following the radiation, you understand all the processes that go through with that, right? So, this idea of using marking insects with radiation is usually a very good way to eliminate a given insect from a region, right? Uh, and now talking more about what can we do with tracers. We can verify, for instance, fertilizing efficiency. You get a fertilizer, and you in your lab you do, or in a controlled environment or some kind, you put some radioactive material inside your fertilizer, and you see if which plants are absorbing that fertilizer, right? And then you can understand, okay, this chemical form of the fertilizer works for what I want, this other one doesn't. You can understand soil fertility doing that, or the way that any kind of fertilizer or something disperses through the land, to understand how soil chemical works. You can have, for instance, to understand plant and animal nutrition, underground water pathways, and all of these only talking about agriculture. When we go to industry, there's the same thing that we can do with underground water pathways. We can do with any type of tubes, for instance, oil pipes, right? Another thing we do a lot is soil tomography. You take a chunk, you take what they call testimony, right? You get a chunk of the soil, and you do a tomography to understand, or if you have an underground pathway of some kind where you can put your source, you can do a tomography of the soil. It's essentially, you can do an X-ray of the soil to see what is the granulometry of the soil, right? To understand the physics of the soil, not only the chemistry. Another thing that we use, for instance, same idea of tomographies or X-rays, we can do with gamma rays, because X-rays are very good for your hand, for your head, but maybe for larger things it will be absorbed anyway. So you need higher energy things, and you use gamma rays. But you can do that, for instance, on trees, to find, for instance, if a tree is rotten on the inside or not, because the most common thing to happen with a tree is for it to rot inside. If you remember your biology lessons way back somewhere, the tree, the part that really lives in the bark of a tree, is the outside. The inside is just dead cells. So if you just if the inside of the wood rots, right, it rots, or insects eat it or something, the tree itself will be still healthy and will not display signals of that, because what is rotten is dead parts. But then it will break, it will fall, it will cause all kinds of trouble. So we can use gamma ray absorption to discover empty parts inside a tree, just like you do with an X-ray, right? And you find out, okay, this tree is rotten on the inside, we have to take it down. Can also find knots, for instance, or whatever, or forget the name. Yeah, you can find insects that live inside the tree or something. Another thing we can do, for instance, and this, of course, you do that on a controlled environment, on a lab farm or something. You can have an insect or something, and you can, because with larger animals, you want to understand the life cycle, dispersion, and everything, mating. What you do, you may have seen on those Discovery channels and everything. What they do, they plug some markers on the larger animals, right? So that they can understand how what are the social habits of those creatures and everything. For insects, that's not that easy to do. So you can, on your control farm, you can, for instance, put some radioactive material in a few of them, and you see how they disperse, you can identify uniquely that one bug, right? And that could help you understand a lot about their society, how they live, how long they live, what they eat, and everything, right? This is also doing for any kind of underground or flying insects. If you get some bait with a radioisotope on it, you can do that, for instance, for ants and whatever, and you discover where the nests, what's the radius of actions, meaning how long from the nest will they go, because usually that's a very limited range, right? And everything, things like this, right? Other things we can do. There's a lot of air pollution study is made by using radiation. Radiation can help you understand pollution in many ways. You can use tracers to understand the dispersion of liquid contaminants in some given soil or something, to see if it will go to underground water pathways. And then you just put a very small amount of radiation, and you see, because radiation can be detected in very, very small amounts. So if you put some amount of radiation here, you can understand where, right? They, on visible pathways, they use a dye. You put red dye or blue dye, and you see red dye is better. You see it dispersing. But sometimes we're talking about underground things we cannot see, so dye will not be useful, and dye has to be in a certain concentration for you to see it. Radiation can be much lower concentration, and you still see it. And there's other types of things we do, like quantifying rare earths and the analysis of contaminants in plastic containers, like a plastic bottle or everything. And there's also one very important thing, and this, our institute is very strong in this field, is the treatment of industrial effluents, so that you treat industrial effluents with electron accelerator, and then you really reduce the biological or chemical toxicity of that, right? Because you break a lot. Now you're not talking nuclear reactions, but you're talking plenty of chemical reactions. Like it's an electron accelerator, it will not cause nuclear reactions, but it will cause a lot of breaking of molecules, and then you can reduce a lot the chemical toxicity of some effluents, so that you can release them in the nature, and you couldn't before, right? And once again, in the industry also, the same idea of radio tracers can be used. As I told you about plants, but there's also other things that can be done. For instance, it's somewhere else, but I know they use it a lot in oil pipes, because sometimes you have to understand if your pipe system, whatever chemical system you have, what is going where, because sometimes you think something is not doing what it should, and then you insert some radioactive material here, and you see how it disperses, and then you say, okay, there must be something blocked here, so let's just open this section, because I guess that's where the blockage is, right? One other thing we do a lot, there's an electron accelerator here in our institute that does that, and there's there are other companies in Brazil that do the same service. You irradiate polymers with an electron beam, right? And by doing that you may ionize molecules, right? And when you do that, you can do two things. If you have a polymer of some kind, it's a long chain. As I told you, long chains tend to be ionized quickly, damaged quickly. But if we do a little less than that in those, you may induce this chain. You may ionize the edge of a chain like this and make it bind to another chain. And then you have a polymer, and you have polymerization. Mean you get a polymer to get even bigger molecules, and then changing a lot its physical characteristics. And that's the other side too. Sometimes you want to degrade a polymer, and then you increase a lot the dose you put in it, and you start breaking these polymers in smaller polymers or in monomers, which are much easier for nature to deal with later, right? So you may aim at the degradation or the enhancement of a polymer. This is done, for instance, especially in electric cables. We have a very long history of collaboration with a manufacturer for electric cables, because when you irradiate them in a certain way, and that's patented concept, but you develop the concept to see which is the ideal rubber to use and how much dose you should put on it. But then you improve a lot the thermal, electrical, and mechanical properties of the insulator, meaning it will insulate more, it will be more resistant to bending, and especially it will be flame-proof, right? Essentially, all flame-proof electric cables were treated by radiation. Then there's polyethylene foams that you can irradiate to increase chemical resistance, thermal, electrical, and acoustic insulation, right? And the capacity, the elasticity and durability, you may irradiate it to reduce specific weight, or to reduce toxicity, or to reduce odor, because odor means you have some what they call aromatic things left in your thing, in your foam. Then you, if you irradiate a bit more, you may transform these aromatic chains in something else, right? It's used in hydrogel membranes, for instance. There was, I don't know how it's as of today, but here in our institute also they used to produce some hydrogel membranes for especially for burns, human burns, right, skin burns, and for surgical protection, because those hydrogel membranes, they can be put around the skin like you would do with a bandage. They are essentially a bandage, but they are a breathing bandage. Microorganisms cannot cross that bandage, but air can, and so air and water. So you don't have all those problems you would have from a regular band in which usually a regular bandage will delay formation of reconstruction of tissue, and will increase the risk of scarification. With hydrogel membranes you don't have that, and so you produce them here in the institute by radiation too. And there's also the thing about non-chemical latex vulcanization, which is important because the traditional way to do latex vulcanization is chemical, and it can leave residue that could be toxic for some people. There's other things very important that people do. There's always lots of sources that can be used, depending. For instance, americium, it emits alpha particles and very low energy gamma rays, so it's for something that you require particle with low range, little short range, right? Most used ones, because they are cheaper also, cesium 137, cobalt 60, and iridium 192, and so on. But what are those things used for in the industry? And now let me tell you, just before that, yes, they are used on daily operation of most traditional industries, right? For instance, every paper company, you produce paper for printers and everything. How do you think they measure the thickness of those papers, right? Because you have 100, you have 60 grams per square centimeter, you have 90 grams per square centimeter, you have 120 squares. There's all this, when you go buy paper for your printer, there's all these choices. And of course, the machines are tuned to give you that kind of paper. But how can you say the machine is doing fine, is working fine? You usually use an americium source, very short, range, and you pass the papers through there, right? Or by sampling or everything. But then you can see exactly which is the attenuation you expect for each of those gauges. And all of a sudden, if something is not going as you expected, you just shut down a part of the machine and say, okay, this requires tuning now. And yes, every paper factory do use that on their daily routine, right? Level gauges also. So you have, let's say, for instance, you have a Coca-Cola factory. Coca-Cola, you want to buy it, and it has to be filled to a certain point. Of course, your all the system is programmed to deliver that amount of soda into the bottle, and the traditional way of doing the safety check, in this case, right, the quality check, was to weight the bottle before and after. But waiting it before and after is hard. You could weight it just after, and then you didn't know if that bottle was heavier or lighter or something. And then you have other issues on the process. And now what they do, they put the bottle and it passes through a radiation source and a detector, right? The concept is the same for many things, is the one that's explained down here. If you have a source here and a detector here, if the liquid, whatever the liquid is below that source, all radiation is reaching the detector, or some of it is absorbed on the walls, but you know how much it is. Once the liquid goes above the source, a lot less radiation will hit the detector, count rate will reduce a lot, and you know that water or any liquid has passed that point, right? So that's what they do with bottles for soda, in many cases for beer and everything, for cans, right? And so it's widely used by food and beverage industry, and it's also the standard essentially for verifying the levels in industrial tanks of any kind. Sometimes you're talking, we could use, on old places we would use just a floater, right? You may have seen those. We used those in home water filters. When this fills, the floater goes up and then it closes the inlet, right? Okay, but or in your car you use a floater. There's a floater on the tank. Once it gets to depending on the position of the floater, you know how much fuel you have on the car tank. But in industry, it's not that simple most of the time. Maybe the liquid you have here is corrosive. Maybe it's just too dense, or very little dense, or everything, and there's a chance it will attack the floater or whatever system is attached to this floater, and it may even be contaminated by what it does to what the remains of the floater or whatever are. So the best way to do, they usually have not only one but sometimes more sources and detectors, because then you say, okay, you have, just as an example, one down here, one up here. If the liquid goes down, you have one here that says, okay, tank is empty, and the other says, okay, tank is full, right? And then you don't need any opening, any visual clue, nothing. The tank is sealed, is perfect, no risk of contamination inside your tank, and you can see in real time the levels, right? There's other things like, for instance, we can use especially heavy particle irradiation of plastic films. You will produce small pores in that membrane, right? If it's very thin, the radiation will essentially leave a small hole there, very, very small hole, and depending on the radiation and on the particle and on the energy, you can tune which is the width of this very precisely. so you can do micro and ultrafiltration with pores from 100 nanometers to 1 micrometer using this. One other thing, and this was done in a nuclear reactor, for instance, you can get a silicon monocrystal and irradiate it in a nuclear reactor. Silicon 30 will interact with a neutron producing silicon 31 that decays to phosphorus 31. And when you do that, you just doped your silicon crystal with phosphorus, which is what you want for circuits, diodes, and everything. The main advantage here is that it's very, very homogeneous, because neutrons cross sections, are small in general. So the whole body of your crystal will be irradiated with the same flux, essentially. So the amount of phosphorus produced in the whole of the body of the bulk of the crystal will be the same, right? You can, for instance, use accelerators, let's say accelerators for heavy ions, okay? You can use them for ion implantation, because remember I told you heavy charged particles, they have a very definite range, and all of them will stop at the same point, right? Depending on the energy, you change the depth, but for a given energy, a given particle, and a given medium, all of them will stop at the same place. So if you want to implant some ions in your, let's say in your diode or something, you need to implant ions. We need that, for instance, for radiation detectors, but it's also very useful for other types of detectors and transistors. And you can implant those ions at a very specific depth by accelerating the ion you want to implant and letting it implant there. Let's remember, what we accelerate are not radioactive ions, right? We accelerate are stable ions. And so you will implant stable ions. If energy is low enough, you won't have nuclear reactions. You will only have the ions penetrating the crystal a certain depth and stopping there. And yes, of course, you have to do an annealing of the crystal, because the path of those ions is completely damaged because of radiation damage, right? And that's essentially what I have to say about uses in industry. Bye bye. Still, I believe 2 more classes to go, yeah, but essentially we're getting there. Hope you're enjoying. Bye bye.